Hi, I'm Juliana, and today I'll be presenting on axon guidance signals. During nervous system development, early neurons must migrate to the correct brain region where they can establish connections with the correct targets. The archetypal adult neuron consists of a cell body and two types of processes: dendrites, which receive synaptic information, and the axon, which transmits synaptic information. During development, the growing axon of each neuron emerges from the cell body of the neuron and must navigate the environment and establish connection with the correct target, which can be anywhere from a millimeter to over a meter away from the cell body of the neuron. So how does each axon find its target in the nervous system with well over 1 billion neurons? To understand how this axon guidance occurs, we first must understand the structure of the growing axon. Here, we can see that there is an axonal shaft made of microtubules stained in green. It extends from the cell body to the most distal end of the growing axon, called the growth cone. As you can see in this image, the growth cone is predominantly composed of actin, stained in red. When we zoom even closer in at the growth cone, we can see that the actin in the growth cone, colored green here, actually interacts with the microtubules, colored purple here, thus directing the growth of the entire axon. Axon bundles can be seen in the protrusions of the growth cone, which are called filopodia. The axon bundles interact with the microtubules of the axonal shaft, and therefore control not only filopodia outgrowth and retraction, but also microtubule extension and direction. Axon is able to retract and extend through a process called treadmilling. The flip end of the axon filament adds ATP-bound globular actin monomers, while the trailing end removes ADP-bound actin monomers. When these processes occur at the same rate, there is no net change in the length of the actin filament. However, if the rate of either the monomer addition or removal dominates, the filament will lengthen or become shorter. During axon guidance, a mechanism for filopodia protrusion involves proteins called clutch proteins. When activated by the membrane-bound protein integrin, stabilize actin filaments and therefore disrupt retrograde flow of actin monomers, resulting in actin filament elongation and filopodia protrusion. The microtubule filaments of the axonal shaft similarly extend during axon guidance through this treadmilling process. In the growth cone, these changes to axon dynamics map to filopodia extension or retraction, and therefore control the direction of growth for the growth cone. Now that we understand the cellular machinery for which the growth cone changes direction, and therefore guides the axon, the question remains of what signal determines whether or not a filopodium retracts or extends, and therefore is responsible for the direction of axon guidance. The signals responsible for axon guidance belong to 4 categories: chemoattractants, chemorepellents, contact attractants, and contact repellents. Filopodia serve as sensors for these signals. One example of cell-bound signaling is contact adhesion molecules, or CAMs. CAMs are used for follower neuron fasciculation with pioneer neurons, a method of axon guidance, and mediate axon-axon interactions. One example of such CAM signaling is with cadherin. The cadherin of the following neuron binds with the cadherin of the pioneer neuron. The actin cytoskeleton of the follower neuron growth cone is stabilized, therefore promoting further axon growth. Another example of cell-bound signaling is ephrins, which are repulsive signals that are heavily involved in the development of the retinotextile system. Ephrins are membrane-bound proteins that are expressed by target neurons, while ephrin receptors are expressed in the growth cone of growing axons. Temporal retinal ganglia axons, which have high ephrin receptor concentration, map to the anterior or rostral side of the superior colliculus, where the ephrin ligand is expressed at a low level on the surface of target neurons. Conversely, nasal retinal ganglia axons, which have low ephrin receptor concentration, map to the posterior or caudal side of the superior colliculus, where the ephrin ligand is expressed at a higher level on the surface of target neurons. This system allows for axon guidance with minimal repulsive signals. Ephrin receptors, which are receptor tyrosine kinases, are thought to cause repulsion due to downregulation of stabilizing cell adhesion proteins, like cadherins. It is also notable that there are multiple types of epherin ligands and receptors. Some epherin ligands are linked to GPIs in the outer leaf of the plasma membrane, while other epherin types are transmembrane. Transmembrane epherin ligands allow for reverse signaling into the tectum. Secreted molecular signals are also critical for axon guidance, which is exemplified through spinal commissural interneuron axon pathfinding, which relies on both attractive and repulsive secreted molecular signals. First, netrin, secreted by the ventral midline, binds to DCC receptors expressed by the growth cone of these commissural axons. This binding activates several downstream signaling pathways in the axons, which ultimately cause cytoskeleton changes that initiate growth cone extension, giving rise to the attractant effect. When the growth cone reaches the midline, and therefore the DCC receptors are oversaturated, the growth cone down-expresses DCC and starts expressing a receptor called ROBO. The ROBO receptor binds to a ligand, SLIT, also secreted by the ventral midline. This binding activates cell pathways that decrease growth cone expression of stabilizing adhesion molecules and inhibit actin polymerization. Therefore, the commissural axons are repelled from the ventral midline, and the crossing of the midline is complete. In summary, axon guidance is a critical component of nervous system development, as it ensures that axons are correctly mapped to their targets, and therefore circuits can form correctly. During development, axons find their target through signals that control the trajectory of axon outgrowth, specifically by altering the cytoskeleton of the growth cone and the axonal sheath to grow in the correct direction. The signals involved in axonal guidance are numerous and complex, as shown through this diagram of human axon guidance signaling, but can be broadly categorized as cell-bound or secreted and attractive or repulsive.